अब हम बात करेंगे डीएनए सीक्वेंसिंग के हवाले से। मॉलिक्यूलर बायोलॉजी में डीएनए सीक्वेंसिंग की ख़ास अहमियत है। जब से ये मेथड डिस्कवर हुआ है और सक्सेसफुली इम्प्लीमेंट हुआ है, इट मेड पॉसिबल कि हम जो फ्रैगमेंट्स ऑफ डीएनए हैं या जीन्स के स्ट्रक्चर्स को एक अरेंज्ड न्यूक्लियोटाइड सीक्वेंस के तौर पे जान सकते हैं, एंड वी कैन कॉल देम एज केमिकल एंटिटीज ऑफ डिफाइंड स्ट्रक्चर्स. All the molecular biologists who are working with the DNA, they must or they sometime have to work with the sequencing, because whenever you are working with something different or you want to work with a new clone, you need to confirm its structure or the sequence of nucleotide, whether this is the right piece of fragment which you started working with or this is something else. आजकल अगर हम देखें आज के दौर में डीएनए सीक्वेंसिंग इज यूज्ड टू कैरेक्टराइज द क्लोन्ड cDNAs. These cDNAs, which are obtained from the reverse transcription process using the mRNA as a starting material, then the cloning is done, and then the cDNA's sequence has to be determined whether this is the same sequence or any changes have been there, or this is not the a specific sequence which we started working with. Then this method is used to confirm or to identify any mutation. Mutation, as you know, is any random change in the sequence of DNA that can lead towards the changes in the structure or the sequence of the protein. So we can determine any mutation in the given gene. We can check the fidelity of newly created mutation or ligation in any DNA mixture. We can sequence the PCR product, and we can decide where and when and how this PCR product can be used further. If we go back a few years, when the automatization was not so common, जो मेजर मेथड्स हैं, स्टेप्स हैं डीएनए सीक्वेंसिंग के, उनको एक मैनुअल तरीके से अंजाम दिया जाता था. और मैनुअल तरीका जो है, दैट इज टाइम कंज्यूमिंग, दैट नीड्स एक्सेसिव लेबर, एंड द मोस्ट इम्पॉर्टेंट थिंग that the manual system or the manual labor was involving number of radioisotopes to be used by the researcher. These radioactive labels could cause or had caused the health hazards to the researchers, but with the passage of time, जैसे जैसे टेक्नोलॉजी एडवांस होती गई, कंप्यूटराइजेशन एडवांस होती गई, हमारे पास अब अवेलेबल हैं ऑटोमेटिक सीक्वेंसर जो मैनुअली हमारा जो लोड है, वर्क लोड है और मेथड्स है, उनको उसने काफ़ी हद तक कम कर दिया है. वी सिंपली हैव टू गिव द सैंपल, एंड दिस ऑटोमेटिक सीक्वेंसर हैज़ ऑल द data, has all the steps performed automatically, and at the end the results are displayed in the form of a computer file which we can study over the computer. The automatic sequencers have decreased the manual labor, and once the manual labor is decreased, it also replaced the use of radioisotopes, so the health hazards are also reduced. And this automatic sequencers, they are equipped with primer synthesizers, and they detect the nucleotides based on fluorescent detection system, and that fluorescence comes out from the fluorescently tagged labels which we use with different nucleotides in the DNA fragment.